So just to show this in a bit more intuitive way, maybe any cell will in your body really will receive thousands of inputs at any given point in time. These can be different things from hormone levels, small compounds, drugs, interactions with other cells in the tissues, so the pressure changes and so on. All of these things will give input to the cell, and the cells have to deal with this in a dynamic way. And the way it does that is to basically calculate changes to its phenotype using, in particular, phosphorylation-based signaling networks or protein networks. And so the goal of our research is really to try to understand how cues alter signals and then how they alter the cellular responses. And the goal is, of course, to use computational models to be able to do this in a forecasting manner, so in a way where you can, in a forward sense, predict how the networks in the cell actually directly correspond to changes to the cellular phenotype. So the way we do this technologically is to sample the genotype or the genome of the cells using high-throughput sequencing in time series studies. We then sequence and monitor the dynamics of proteins, so the concentration of proteins on a global scale within the cell, and also how the interactions between these might be changed, particularly through phosphorylation-based changes to the proteins. And then at the same time, we do a lot of high-throughput imaging. And it's really this part of the pipeline, if you will, I will spend most of our time today about, is really the imaging side of things and how we try to correlate and develop deep learning models for doing the forecasting into these complex biological spaces. And of course, we don't only look at a single cell level, we also study multicellular systems, in particular tissues and animal models and also patient samples to get much more complexity in and what we call multiscale studies, so we can start challenging whatever computational model we have developed in much more complex and much more realistic and relevant biological setting, in particular from a disease point of view. So that's essentially the pipeline, if you will. So the way this operates is eventually that we perturb the cells, expose them to a huge number of challenges, and this goes into a deep learning pipeline, which I will discuss later. And then at the same time, we are monitoring basically how the networks in the cells are being altered. And the three technologies we use for this is high-throughput imaging, shown here, robotic imaging. So this is automated laboratory, basically, that performs huge number of individual experiments using FDA-approved drugs, genetic perturbations, RNAi, CRISPR, also natural compounds and so on, is basically being conducted at the rate of hundreds of thousands of experiments, individual experiments on individual cells or on collections of cells. And then you're doing basically high-throughput imaging to see how the cells are changing their shape and their behavior. At the same time, we're monitoring the cellular dynamics in terms of molecules and particular protein dynamics using mass spectrometry. This is Orbitrap technology. So basically a Fourier transform-based type of mass spectrometry where we can measure very accurately at the global level changes to proteins and modifications of proteins. In the center of all this is really the supercomputing. So this gives rise, and I will show this in a moment, to very large data sets. And there's just no way you can handle this without not only high-performance computing, but actually in our case, absolute requirement for large shared memory systems, and in particular, the new types of shared memory systems. We have monolithic systems from SGI has been key for dealing with this kind of data structures. One of the things I want to point out is, of course, one should be aware there's always a whole array of different types of modeling approaches. Deep learning is one of them. The ones I feel is very important for biology because we don't always know atomic details in the system, but as you know, there's many other types of modeling approaches, Boolean models, statistical models, and so on. And it really depends on your question and your data at hand, which ones you want to use. So one of the key aspects, though, is that to be able to explore different types of models, it's very important to have large computational dynamic range. And by that, I mean be able to scale out and scale up and scale into memory space without having to necessarily write a billion lines of code of MPI code, but just do it freely. And move between these different paradigms of computing is critical for having a freedom in your computational modeling, so you don't pre-select different models only based on what you can do with your hardware. So this is actually very key and one of the main drivers for sort of our collaboration with SGI.